Speaker, Riekelt Houtkooper, who I am, and he doesn't know that, but I will claim it. I'm a big fan of what he's done previously in the NAD field. This is what really inspired me, and I think a lot of other people, to try to understand how NAD could impact aging. Riekelt, can you hear me?
Yes, I can hear you. Can you hear me as well?
We can hear you, and so we're very excited to hear your talk, which I guess will be a little bit about mitochondria and longevity.
Exactly, yes, mitochondria, not so much NAD. But first, I would like to start by congratulating the organizers on a magnificent meeting. I think it's a really wonderful and interesting combination of talks on various topics of aging and longevity. Yeah, the work, let me first put on the right pointer. The work that we do in my group is centered on metabolism and various aspects of metabolism and how it interferes with ultimately the aging process. And we try to understand mainly two factors. On one hand, genes and regulation of genes and how it influences this metabolic network, how it tweaks certain bypasses in metabolism, for instance. And on the other hand, nutrition and nutritional factors that feed into this metabolic network and need to be converted into energy or building blocks for cellular growth. And like I said, in the end, we would like to understand how these metabolic changes lead to what I call here metabolic health and aging. And for a long time, I've been interested in these kind of complex networks of longevity, how these metabolic pathways intersect, how caloric excess and caloric restriction pathways converge and ultimately lead to activation or inactivation of either anabolic or catabolic pathways and thereby lead to a state of longevity. And when we started doing these kind of experiments, one of the first studies that I did is quite a big systems genetics approach using DXD mice. It's a population of different wild-type strains of mice, shown here on the left. Each dot here represents one strain of mice. And we found that the expression of a certain gene, mitochondrial ribosomal protein S5, correlated very strongly with the lifespan of these mice. And so this is a natural population, similar in terms of genetic variation with human population. Obviously, the correlation in mice does not necessarily reflect causation. And so we moved to C. elegans, where we could silence the equivalent gene, MRPS5, to see if this would indeed support our idea that this was a causative gene for lifespan extension. And indeed, we saw this 60% extension in lifespan. And this was not only the case for MRPS5, which was the first gene we identified in the pathway, but actually also for other family members of MRPs, and I will come back to that in a second. Because what do? These MRP proteins, mitochondrial ribosomal proteins, are actually, it's a family of 70 to 80 different proteins that constitute the ribosome within mitochondria. As you probably all know from your second year in university, most of the genes in mitochondria, or most of the genes anyway, are encoded in the nucleus. They are translated, and for instance, these oxidative phosphorylation subunits, they are imported into mitochondria, into these complexes 1, 2, 3, 4, and 5 of the respiratory chain, or oxidative phosphorylation. But 13 protein coding genes are actually encoded by the circular mitochondrial DNA. They have a separate transcription and translation machinery, of which MRP are a major factor. They synthesize these genes, and they are also incorporated in the oxidative phosphorylation machinery, the complexes, and particularly complex 1, 3, 4, and 5. So this is what MRPs do. And of course, we wanted to know a little bit more of the mechanism. Why is silencing one of these genes sufficient to extend lifespan? And we were helped a lot by a paper that just appeared at that time from Andy Dillon's lab, where he showed that inhibiting certain mitochondrial respiration functions initiates a cascade, a protective cascade of mitochondrial unfolded protein response. basically, to run you through this, what happens, what initially signals the cascade is an accumulation of unfolded proteins. They are cleaved into peptides, exported through this Have1 peptide transporter. And this initiates a cascade of transcription factors and transcriptional co-activators that together induce the expression of heat shock proteins, chaperones, and proteases that travel back to mitochondria and restore proteostasis there. And so we tested if our modification, our MRPS5 RNAI, indeed led to an activation of this pathway, and whether it could explain the lifespan extension. And indeed, we used a worm model where HSP6 is one of the marker genes for the unfolded protein response, the UPRM. And we tested if this is activated, and indeed, in the condition where MRPS5 is knocked down, we see a massive activation of unfolded protein response, the mitochondrial unfolded protein response. What we also saw, and we didn't pay so much attention at the time, is that if you look at the morphology of mitochondria, and we do that with a worm strain that expresses GFP and it's targeted to the mitochondria in the muscle, we see that under normal conditions, we have this sort of tubular network kind of structure of mitochondria. So this is the actual image, and this is a reconstruction. And if we then silence MRPS5, we see that rather than this tubular network, we see a more fragmented state of mitochondria. And also, if you look at the EM images, for instance, here you see these long tubular mitochondria, and they are totally gone in the context of MRPS5 RNAI. These mitochondria are more circular and smaller. Like I said, we didn't pay too much attention at the time the paper was published, and only a couple of years later, Jasmine Liu is a PhD student, almost about to graduate, and she's actually looking for postdoc opportunities. So if you're hiring, I would certainly recommend her. She asked me the question. So basically she said, okay, it's all nice that you find this MRPS5 extending lifespan, but you also find this fragmentation of mitochondria. But what does it mean? Is that somehow functionally involved in the lifespan extension that you observed? And I had to say, I didn't know. We never tested that. And so she set out to test it. And it's important first to describe a little bit of molecular biology of mitochondrial dynamics, because we are starting to interfere in the next couple of slides with some of these processes. So mitochondrial fusion is managed by a couple of mitochondrial fusion proteins, especially these mitofusins, which are involved in the fusion of the outer mitochondrial membrane, and OPA1, which is involved in fusion of the mitochondrial inner membrane. On the other hand, mitochondrial fission has a lot of factors involved, including ER that engulfs or encircles basically these mitochondria, but especially the one that I want you to focus on is DRP1, which is the main fission factor for the rest of the story. It induces the fission. And in worms, actually, these are called FISO1, EAT3, and DRIP1. And I will keep referring to both the mammalian and the worm name throughout. And so the first experiment that Jasmine performed was to silence fusion and to enable fission to occur. So you would expect a more fragmented network. And indeed, where you again see in the control condition, this tubular network of long stretches of mitochondria, if you inhibit MRPS5 and you inhibit also EAT3, so the OPA1 homolog, you see again the circular kind of mitochondria. So what happens to lifespan? First of all, the empty vector is the regular lifespan. These worms live about 30 days. MRPS5, like I showed before, they have a lifespan extension, the purple line. And if you now silence MRPS5 and EAT3 at the same time, you see the synergistic effect of enhanced fragmentation, enhanced fission, plus inhibiting mitochondrial translation. Then, of course, the next step was to do the reverse reaction. So we block fission and thereby promote fusion, enhancing basically this mitochondrial network state into a tubular network. So what happens then? And our initial expectation was that the reverse would be happening, but that turned out to be incorrect. So under these conditions, actually, again, we have the normal N2 control line and the MRPS5 here in purple. And if we now delete DRIP1, the fission factor, so we promote fusion, and then we apply this MRPS5 RNAI, we see even a further extension, similar to what we saw when we promoted fission. So this was quite confusing. And we wanted to understand what is going on here. And one elegant experiment that Jasmine came up with and that we could do because of the worm strains that were generated in Will Mears' lab is use a worm strain that has both fusion and fission knocked out at the same time. So this is a DRIP1, FISL1 double mutant, which basically means the mitochondrial network is frozen. And you can see instead of MRPS5 being able to induce some sort of fragmentation, in this case, several markers of mitochondrial morphology remain exactly the same despite the treatment that we unleash on these worms. So the mitochondrial network is indeed frozen. What does it do to lifespan? And this is actually where we see that the dynamic network of mitochondrial function is required for the lifespan extension of MRPS5. So under these conditions, MRPS5 in the wild type actually extends the lifespan, but in the double mutant where the network is frozen, in fact, it doesn't extend, it even shortens a little bit, suggesting that the dynamics is required. So that's my first set of conclusions, is that blocking either fusion or fission is actually beneficial and synergizes with this reduced mitochondrial translation to extend lifespan, in contrary to our expectation. However, the simultaneous disruption of fission and fusion, thereby freezing this mitochondrial network, completely blocks the MRPS5 RNAI-induced fragmentation and lifespan extension. Well, I showed you in the beginning that in our previous work, we found that there was an important role for unfolded protein response in managing these lifespan extending effects of MRPS5 RNAI. And so the question came up, if this was actually also the case under these conditions, is UPR somehow involved? So the first thing we tested is under these various conditions that I just showed you, where we see either synergistic lifespan extension or no lifespan extension anymore, is there still activation of UPR? So we tested the expression of HSP6, and indeed, as we showed before, MRPS5 activates UPR, but also under the conditions where we block fission or where we freeze the mitochondrial network, in both those conditions, we still see activation of unfolded protein response. And if we then use a mutant for HAF1, which is the peptide exporter that is important for the initiation of the unfolded protein response, we actually see that still this dashed line, there is an extension or synergistic extension of lifespan when we inhibit MRPS5 and E3. Suggesting that UPR is not involved in this process. But what is? So to answer this question, Jasmine started doing basically a small-scale screen for transcriptional regulators and different kind of regulators of lifespan, hoping that she would find one that would abrogate this lifespan extension of MRPS5 and bring it back to the empty vector control. And indeed, after screening all these different genes, only one appeared to be positive, or one was positive, HLH30, shown here on the right. So this is an E3 mutant in which we block MRPS5, basically the purple line. So this is again the synergistic lifespan extension that we showed before. And then if you silence HLH30 in this particular context, you see that it brings it back to the control lines, confirming that HLH30 is important for this lifespan extension. And the same is actually true for the fission mutant, the DRIP1 mutant. So what is HLH30? It's actually the worm homologue for TFEB, a transcription factor that regulates lysosome biogenesis and autophagy. So under normal conditions, TFEB resides mostly in the cytosol, and upon activation, it travels to the nucleus, activates gene expression, to initiate all sorts of processes related to lysosome biogenesis and autophagy, as you can see here. So the first thing we wanted to test is if HLH30 was indeed going to the nucleus. And this is the empty vector control conditions. You see that the GFP, which is coupled to HLH30, is sort of a faint distribution across the cells. Upon silencing MRPS5 alone, you already see these structures appearing, which is nuclei accumulating HLH30. But this is amplified very much so when we silence MRPS5 plus E3. So suggesting that in this condition of amplified lifespan, we also see an amplified HLH30 translocation. And this is quantified here, where you see the dark red, really more HLH30 nuclear localization compared to the other conditions. Does this also lead to changes in the lysosome? So we did EM. I'm skipping here some of the control conditions, but I hope you believe me when I tell you that these kind of structures, shown also here in more detail below, these multivesicular bodies and electron-dense structures, indicating sort of lysosomal biogenesis and activation, are more prevalent in the E3 mutant that was silenced with MRPS5, so the amplified lifespan extension, and that we could strongly attenuate this when we silenced HLH30 again. So you see a disappearance of all these kind of structures, confirming that HLH30 is driving this phenotype. And then the last question we answered is this also leading to an increase in autophagic flux, because that's the ultimate downstream effect that we would like to see. And here we used a reporter that basically is a dual reporter in which the autophagosome is stained yellow, and autolysosome, the terminal endpoint of this autophagic flux, is stained red. And particularly in the context where we co-silence MRPS5 and E3, you see the appearance of all these autolysosomal structures in these worms, confirming that indeed the downstream endpoint of this pathway, increased autophagic flux, is met. So to conclude, we found that under these conditions, UPRMT is actually not the driving factor of the lifespan extension. In this intervention where we inhibit both mitochondrial function and form. It's actually HLH30 or TFEB that mediates the lifespan extension. And the double RNAI of MRPS5 E3 promotes the nuclear localization of HLH30, stimulates mitochondrial lysosomal biogenesis, autophagic flux, and drives the longevity phenotype. Again, this is modeled here. So under normal conditions, fusion and fission are in balance. This remains actually HLH30 in the cytosol and leads to a normal lifespan. But if we inhibit mitochondrial translation and we co-inhibit either fusion or in the bottom left, the fission, we see either fragmentation or enhanced fusion. And both scenarios lead to a synergistic stimulation of HLH30 going to the nucleus, driving this lysosome biogenesis, autophagy, and promoting longevity, which is totally blocked if we silence both fission and fusion, and we don't see lifespan extension anymore. To close, I would like to take the opportunity to point you to the talk of Marta Molenaars, another PhD student in my group, also wrapping up her PhD and soon defending and actually looking for a postdoc opportunity in New York City. So I encourage everyone in New York City to listen to the talk and approach her. She will present a story that was recently published in Cell Metabolism that basically links to this work as well. And with that, I would like to thank the people who were involved in this work, particularly Jasmine, Rebecca, George, and Alison, and the rest of the team in Amsterdam. And also our collaborators in the imaging facility, ETH Zurich for the proteomics that actually I didn't show, Palas and Will for the strains, but also for some complicated imaging that they did for us. And finally, the financial support that allows us to do all this work. And with that, I'd be happy to take your questions.
Thank you very much, Riekelt. Really great talk. We have a number of questions online. The first one is from Attila Cortes, I think it's pronounced. He's asking, have you done any experiment related to mitochondrial transfer between young healthy donors and older compromised recipients, or the other way around?
No, we haven't done those kind of experiments. Sounds cool, but we haven't.
That was a great short answer. I appreciate that. And I have a question then. Fantastic talk, Riekelt. What is the redundancies and overlap between the different UPRs? And would it be beneficial to activate all of them or be more selective in terms of health and longevity?
Yeah, it's a great question. I wouldn't activate all of them at the same time. I think they're there for a reason and they shouldn't be overactivated. Also, I wouldn't argue that activation of UPR, whether it's MT or in the ER, is always good. We've seen also several examples in our own studies where activation of UPR is actually associated with shorter lifespan. I have to say that under these conditions in this study also, we tested also ER UPR, for instance, is not activated. So I think all those UPRs are extremely time and context dependent pathways, and I wouldn't advocate for activating all of them massively.
I see. Okay, I think we will stop here in the context of time, but really appreciate your talk, fantastic, and please go online on Slack and answer the questions that are there. And then we have...
I will, thank you so much.
Thank you, Riekelt.